Okay, let's talk about glycolysis. Now I'm going to do this glycolysis a little bit differently because this is a medical class. I will be leaving out structures and just referring to the compounds that are made and the enzymes that mediate these changes. So the first thing we need to talk about is insulin, because we are in a fed state in the beginnings of our discussion. So the insulin on skeletal muscle will induce the translocation of GLUT4 to the cell surface of skeletal muscle. And if we talk also, I want to have a little discussion about red blood cells. Red blood cells are going to be using GLUT1, which is another high affinity glucose transporter, as is GLUT4, but GLUT1 is not responsive to insulin. So this is on the cell surface of red blood cells all the time. And this makes sense because red blood cells need to perform glycolysis at all times to generate ATP, whereas skeletal muscle is going to generate ATP and store glucose in the fed state, and thus it's responsive to insulin. So now once we have the glucose entering the cell in both conditions, the glucose is going to be phosphorylated to glucose 6-phosphate. This is going to be using the energy of ATP, and the enzyme to do this, of course, in the skeletal muscle is hexokinase. It is also the same enzyme that is used in red blood cells. So all we're doing here is this is part of what's called the preparatory phase of glycolysis. This is going to be preparing the glucose by using ATP and adding a phosphate on the number 6 carbon. Not only preparing the glucose for the subsequent reactions in glycolysis, but also to prevent glucose from exiting the cell through the transporter, because glucose 6-phosphate is not recognized by the transporter. Now, regulation of this particular step is done by a negative feedback inhibition. So the product, glucose 6-phosphate, binds to its enzyme, hexokinase, and inhibits. This type of inhibition is not necessarily that important in skeletal muscle. Of course, not very important in red blood cells. However, this will become very important in the liver. The next step in glycolysis is an isomerization reaction. And we are simply shifting the aldose form of our hexose glucose 6-phosphate to the ketose form. This enzyme is phosphoglucoisomerase. The isomerase enzyme simply converts the functional groups aldehyde into a ketone. Important to note that this reaction with hexokinase is irreversible because it harnesses the power of ATP to drive the reaction. However, this reaction here, as we'll see later, is reversible. The next step in glycolysis is going to be another phosphorylation reaction, part of the preparatory phase. And here again we will use ATP to make fructose 1,6-bisphosphate. The enzyme that does this is abbreviated PFK1. We will be looking at a lot about this enzyme, which is phosphofructokinase 1. This enzyme is regulated in muscle by ATP levels. So ATP, as ATP levels rise, this will be an inhibitor of PFK1. And this makes sense because as the skeletal muscle recharges itself to fully increase its capacity of ATP, you want to slow this glycolysis down, and we'll talk about why in a subsequent video. The inverse of this is also true. AMP, which is the energy depleted form of ATP, is also an activator of PFK1. So as skeletal muscle depletes its ATP levels and AMP starts to rise, this will activate PFK1 and increase glycolysis. Now fructose 1,6-bisphosphate at this point will be converted into two separate products. We are going to take our 6-carbon intermediate, and we are going to make now two 3-carbon intermediates. The first one is dihydroxy acetone phosphate, abbreviated DHAP, and the second product is glyceraldehyde 3-phosphate. The enzyme that performs this reaction is called aldolase, and simply cleaving this substrate, fructose 1,6-bisphosphate, right in the center to produce two 3-carbon intermediates. Now the rest of glycolysis is going to be picking up from glyceraldehyde 3-phosphate, and so we need another isomerization reaction to convert DHAP into glyceraldehyde 3-phosphate. And this reaction is being done by an enzyme called triose isomerase. So it's simply an isomerization reaction to take the ketose form of the 3-carbon sugar and return it to the aldose form. So going from a ketone to an aldehyde. Now the glyceraldehyde 3-phosphate is going to be processed next into 1,3-bisphosphoglycerate. This enzyme is done by glyceraldehyde 3-phosphate dehydrogenase. And this is a dual reaction that is going to add inorganic phosphate to the number 1 carbon of glyceraldehyde 3-phosphate, and also harness some energy by converting NAD to NADH. So it's a combined substrate level phosphorylation reaction with a redox reaction. Now at this point, red blood cells can take a little bit of a detour and convert this to 2,3-bisphosphoglycerate, more commonly known as 2,3-BPG. This, of course, is a small molecule that increases oxygen release from hemoglobin. So as we increase our altitude and we need more oxygen, the synthesis of the enzyme, which is bisphosphoglycerate isomerase, the synthesis of this enzyme increases at high altitude, increasing 2,3-PBG in the red blood cell, which increases oxygen release from hemoglobin. Now, both in skeletal muscle and red blood cells, we have our first ATP generating step where we are going to be forming 3-phosphoglycerate and this is with the enzyme phosphoglycerate kinase. So up to this point, we've used 2-ATP to prepare the glucose molecule for subsequent reactions, and this is our first step where we are going to be harnessing some energy. So we will be generating ATP from ADP. The 3-phosphoglycerate is converted to 2-phosphoglycerate, and this is done by the phosphoglycerate mutase enzyme. Now contrast this enzyme with the isomerase. The isomerase is changing a functional group from either an aldehyde to ketone or ketone to aldehyde. A mutase is simply moving a phosphate group from one portion of a molecule to the other. The next step is to generate phosphoenol pyruvate. The enzyme that does this is called enolase, and this is simply a dehydration reaction, removal of water, to make phosphoenol pyruvate, which is abbreviated PEP. And this is a high-energy molecule, so removing water from 2-phosphoglycerate essentially charges that phosphate group on the pyruvate backbone so that we are able to harness our last ATP from glycolysis using the enzyme called pyruvate kinase and converting ADP to ATP. So we've used 1 ATP to prepare the glucose into glucose 6-phosphate. We've used a second ATP to phosphorylate fructose 6-phosphate by PFK. Now we're starting to harness energy with phosphoglycerate kinase, ATP released, and pyruvate kinase also generating ATP. We have 1 hexose going into the reactions. At this point, after the preparative phase, we have 2 3-carbon molecules continuing, and so these ATPs we multiply by 2. So we have a net gain of 2 ATPs going from glucose to pyruvate. Now the fate of pyruvate we'll talk about when we talk about the TCA cycle. We can process pyruvate into acetyl-CoA and enter that into the TCA cycle to generate more energy. Or, if we happen to be in an anaerobic state of respiration, which is lack of oxygen, we would be in this state in skeletal muscle if we've surpassed our oxygen delivery mechanism to our muscles and our muscles go anaerobic because of this lack of oxygen, or all the time in red blood cells, because red blood cells do not have mitochondria and so they cannot perform oxidative phosphorylation. So once we go anaerobic, we can take the pyruvate and convert this into lactate. The purpose of this is to regenerate NAD from NADH. So this is a redox reaction by the enzyme lactate dehydrogenase. In this redox reaction, we are going to be reducing pyruvate, oxidizing NADH to NAD to create lactate. The reason we need the NAD regenerated is because we're in an anaerobic situation, and this enzyme here, glyceraldehyde 3-phosphate dehydrogenase, needs NAD to continue working. So in an anaerobic state, if we don't have this reaction of pyruvate to lactate, we will eventually deplete the amount of NAD and stop glycolysis at this level of glyceraldehyde 3-phosphate dehydrogenase. So at this point, the lactate can be secreted into the blood, and tissues can pick this up. But what we are going to talk about primarily, and as we move along, is that lactate is a very important substrate for gluconeogenesis in the liver.